Good day. I am Eliza May De Leon Abulag, a fourth year BS Marine Biology, and my study talks about carbon stock evaluation of a mangrove ecosystem in three selected barangays in General Santos City. These are the content of the report. First, introduction. Second, methodology. Third, results and discussion. Fourth, conclusions and recommendations. Mangrove forest provides a variety of ecological goods and services, as well as serve as key carbon sink in the tropics. Despite this, mangrove conversion continue. Carbon stock in biomass and soil, as well as soil emissions of greenhouse gases, are critical inputs for analyzing the impact of mangrove conversion on the carbon budget. Blue carbon refers to carbon stored or sequestered in marine and coastal ecosystems, including mangrove forests, tidal salt marshes, and seagrass meadows, as well as coral reef and oceanic carbon sink in the form of marine algae. These are the main objectives to evaluate carbon stock in mangrove forests, specifically in General Santos City. First, assess the vegetation parameters like ground cover, health status of the mangroves, and diversity indices. And B, assess the above ground and below ground carbon stock and the total carbon stock. Carbon stock evaluation is an important topic in global carbon cycle. Total forest carbon stock mainly includes vegetation, soil and litter, carbon stock, an important parameter in sequestering capacity and carbon budgets. The findings of this study will help the community, local government, volunteers, and sectors in restoring mangrove forests by promoting knowledgeable policy, informations, and instruction in regard to mangroves conservation. For the future researchers, which aims to compare and contrast the presence of carbon stock information regarding mangrove forests in any preferred places. It will also provide informations, guidelines, and protocols to assess carbon stock in the future assessment. Interested student researcher will also find this study as the references to also evaluate the assessment for the significance, abundance, comparison, and detailed information on the mangrove's health in the ecosystem. It is important also to know the scope and limitation of the study. The study used quantitative sampling method to evaluate carbon stock in General Santos City in determining species diversity and carbon stock in above ground and below ground pools of mangrove forests. During evaluation, particularly in Barangay Baluan and Buayan, preliminary conduction happened in the study site, which is General Santos City, South Cotabato, on the day July 14, 2021. Using global positioning system, it determined the specific coordinates. The total of 8 overall coordinates had been collected on the total carbon stock from above ground and below ground plant and used the sampling method by Kaufman in 2011. Due to some instances, the mangrove percentage was limited into 10% of the total area cover in mangrove forests because of the unevenness distribution of the sampling coordinates. A random or clustering sampling has been used for the purpose of quantification of acidity and qualification of carbon stock in mangrove forests as well as the internal given. These coordinates were plotted and marked during preliminary assessment for sampling location and mangrove area covered. An estimated 10% of the total mangrove area, together with 15 by 15 meters interval, had been assessed and measured. In vegetation analysis field assessment, these four parts are the important. First, select project area boundaries. It is important to know your boundaries, especially when identifying your project area. Second, decide stratification of the area, or you have to know the arrangement, classification, or the status of the area given. Third, decide which carbon pool to measure. It is important to know which carbon pool you are going to measure, to know your small scale or broad scale boundaries. And lastly, determine type, number, and location of measurement plan. It is also important to know the type of measurement you're going to use, number of plots you're going to measure, and location of the measurement plot. Somehow, due to some instances, we have our different conditions in measuring the diameter breast height of the mangroves. These are the following, and we have to use the different conditions by English et al. In 1997. These equations are used to calculate species importance in terms of vegetation parameters. Based on Shannon and Weiner, diversity indices were used in the determined quantitative description of mangrove habitat in terms of species distribution, and evenness based on Shannon-Weiner index, where H index pi proportion of important values of species I to the total values of all species, and in the natural logarithm, while Shannon-Weaver's species distribution evenness, pi the relative abundance of each group of organisms. The values are rated based on the Fernando scale 1998. Regeneration and percent cover measures to determine the condition of the mangrove ecosystem. Regeneration of saplings, seedlings, and trees was computed based on Diggett et al. In 2004. To solve regeneration per meter squared is equal to total regeneration count over total regeneration plots. Crown cover is equal to 0.7854 times d squared, where d is the total of crown diameter. Percent crown cover is equal to total crown cover over the total area sample multiplied by 100. Good day! This is the habitat criteria rating chart of mangroves. If the condition is excellent, We have 76% and above in percent ground cover, 1 regeneration per meter squared, undisturbed to negligible disturbance. If the condition is good, 51% to 75% ground cover, less than 1 to 0.76 regeneration per meter square, slight disturbance and few cuttings remain. If the condition is fair, 26% to 50% ground cover, 0.50 to 0.75 regeneration per meter square, and we have moderate disturbance and noticeable cuttings. If the condition is poor, we have 0 to 25% ground cover, less than 0.50 regeneration per meter square, heavy disturbance, cuttings, pollutions, rampant conversions to other uses nearly destroyed. This is the codes used to describe the type of impact on a site. If the code is CO, it means infrastructure including piggeries, garbage dumps, and developments. If it is ER, erosion occurs, shown by uneven mud surfaces or little scarps. If it is EC or BS, extensive cutting or bark stripping. If it's MI, mining activities such as sand collection. MU, multiple impacts. Note codes of multiple impacts in the remarks. And lastly, OT, others or example of pig foraging. Note this also in the remarks. Welcome to carbon stock data collections for the above ground and below ground biomass. A schematic of plot layout. This design was used for assessing directional gradient, reducing species contagion, and increasing precision for mangrove sample. In a soil carbon sampling based to Kaufman and Donato in 2012, accurately measure the soil carbon with 3 parameters were needed. First is soil depth, second soil bulk density, and the third organic carbon concentration. In the study, samples were collected at 0 to 30 cm, 30 to 60 cm, and 60 to 100 cm depth with the use of metal or improvised corer. 1 kilogram of soil from 3 depths were gathered. Samples were air-dried, labeled, and delivered to near soil and water laboratory center. The methods used to quantify the carbon content uses ultraviolet spectrometer. Let's talk about carbon stock analysis. According to Kumiyama, estimation of above-ground biomass and below-ground biomass were calculated using the allometric equation for mangrove forest. The following allometric equation was used. We have W top is equal to 0.251 PD 2.46. We have WR is equal to 0.199 P 0.899 D 2.22. For soil and carbon pools calculation, bulk density was determined by dividing the oven-dry soil sample by the volume of the samples. This method was already used in the soil laboratory analysis. The equation is as follows. On the other hand, the soil carbon biomass per sample depth interval was calculated. Soil carbon megagrams per hectare is equal to bulk density times soil depth interval by centimeter multiplied by percent of carbon, where percent carbon is the carbon concentration expressed as a whole number. After solving, we found the total carbon stock. The total carbon stock or pool was estimated by adding all the component pools. First, each component pools was averaged across all plots. This average were summed to obtain the total carbon stock according to Kaufman and Donato in 2012. So the total carbon stock, megagrams per hectare of the sample stand, is equal to the carbon of the above ground, add to the carbon of the below ground, plus the carbon of the dead tree, plus the carbon of the woody debris, plus the carbon of the soil. Let's move on to the results and discussions of vegetation analysis and carbon stock analysis. In the study, in General Santos City, specifically in three barangays, which is Bula, Baluan, and Buayan, these are the mangrove species collected. In the family Rhizophoriaceae, we collected Rhizophora macronata, Rhizophora apiculata, Rhizophora stylosa, while in family Abyssiniaceae, we found out Abyssinia marina. In Soneraceae, we found out Soneracia alba, and lastly in Meranaceae, we found out Aegisiras floridum. Next to the criteria is the vegetation analysis of mangrove forests in General Santos City, South Cotabato. Based on the table, Rhizophora macronata had the highest importance value with 117.27, while the lowest is the Rhizophora stylosa with 1.09 importance value. This is the total regeneration count according to seedling, sapling, and trees counted. Based on the table, the seedling count all in all is 81, the sapling count all in all is 115, and the trees count on all in all is 181 counts. Based on the table and computed on its formula, the condition of the seedling and sapling count was result into poor condition. All in all, the crown cover was the was being averaged by the total crown diameter of the species. So the result showed that the whole species collected fall into fair condition based on the habit criteria rating chart for mangroves. It also showed the status of the mangroves gathered fall into poor condition and the type code fall into highly impacted. According to Fernando's scale, results in both diversity evenness being in very poor conditions. Results have shown that mangrove species or the species evenness was 1.6, while the species diversity is at 1.8. The total above ground carbon stock in the sampling plot was 159.25 mean carbon forest density, with live trees accounting for 159.25 mean forest carbon density and the dead downwood accounting for 0.35 carbon density. The mean carbon stock of the sampling area are ranged from 118.23 mean forest carbon density to 200.33 mean forest carbon density in live trees, and from 1.6 E negative 6 to 5.2 E negative 6 megagrams per hectare in wood debris. In tree with large girth diameter and high species wood density, such as Rhizophora apiculata with 34% and Rhizophora macronata, considerable level of carbon were found in the sampling plot with 33%. The total below-ground carbon stock includes the complete amount of below-ground plant and soil carbon pools. Carbon reserves in below-ground plant are an average of 238.19 mean forest carbon density. The mean carbon stock in the sampling plot ranged from 167.99 to 308.39 megagrams per hectare. A total of 24 soil samples were obtained at the sampling plots and categorized by depth, from 0 to 30 centimeters, 30 to 60 centimeters, and 60 to 100 centimeters. Organic matter levels differ by soil depth. 0 to 30 centimeter yielded 33.11 mean forest carbon density, 30 to 60 centimeter yielded 27.32 mean forest carbon density, and 60 to 100 centimeter yielded 27.27 mean forest carbon density. The samples had 87.71 mean forest carbon density in total, with a mean carbon stock ranging from 51.07 mean forest carbon density to 124.51 mean forest carbon density. The total ecosystem carbon stock in General Santos City, South Cotabato, mean carbon stock of 485.18 mean forest carbon density, ranging from 281.98 mean forest carbon density to 688.38 mean forest carbon density, and a mean carbon dioxide equivalent of 1,780.61 mean forest carbon density. The pools from above ground and below ground contributed to the results. The total ecosystem carbon stock in four components have been tabulated. Below ground carbon stock accounted for 49% of the total percentage, whereas above ground carbon stock accounted for 33%. On the other hand, soil accounted for 18% of total carbon percentage, and wood waste from below ground plant has 0% equivalent. Let's move on to the discussion. Species composition, distribution, and diversity and status rate. Mangroves are important ecosystem of tropic zones providing ecosystem services such as sources of food, wood, and protection from extreme events such as cyclones and tsunamis. In the study, 6 species were found in General Santos City, South Cotabato, namely Rhizophora macronata, Rhizophora apiculata, and Rhizophora stylosa, Abyssinia marina, Sunraysia alba, and Aegisiras floridum. It was dominated by Rhizophora macronata with the highest important value in the vegetation parameters. According to Ken Fern, Rhizophora macronata is slow-growing but much-branded evergreen tree growing up to 27 meters tall. with a bole 50 to 70 centimeter in diameter. The tree produces numerous stilt roots from the baseline. By the coast in brackish, saline areas and estuaries, those species are mainly their habitat. Also on the low coastal areas, flooded by normal and daily high tides. The counted regeneration and percent crown cover of species was observed in Table 10, which is expressed in the criteria given. Results showed that the species' total regeneration count was a fair condition, which means the moderate disturbances and noticeable cuttings happen at the assessment site. The code of impact pressure in a site is highly impact. It's because only a few remain at canopy height, other extensive clearances, and some recruitment and garbage dump, and there is a large of bare mud. In the study, the area had been wrapped up by muddy sandy type of soil which made up the presiding of Rhizopora macronata. According to Mangrove Science Database, coastal development activities such as deforestation, dump site, and manufactured into commercialized. To address the country's catastrophic rate of mangrove deforestation and degradation, numerous projects and undertakings related to mangrove restoration and conservation have been initiated. The diversity of a mangrove fall into very poor relative values which correlated to the type impact expressed in the species crown cover. The reason why it became very low is because the diversity and countability of the mangrove collected specifically, the evenness and distribution are not counterparts. Some plots are not even in species countability. General Santos City, South Cotabato's result in above-ground biomass was higher than the above-ground biomass in the municipality of Maitom according to Cinco 2018, and above-ground carbon stock amounting to 241.527 mean forest carbon density in municipality of Maasim according to Bigsang in 2016, but lower in below-ground biomass compared to below-ground biomass obtained in Maitom, Sarangani Province. In addition to that, General Santos City hold a mean carbon stock with 485.18 mean forest carbon density with a mean equivalent of carbon dioxide equivalent 1780.61 mean forest carbon density. Compared to other carbon stock, Maitom Sarangani Province hold 367.89 mean forest carbon density with a mean value of carbon dioxide equivalent of 1350.17 mean forest carbon density, and to the mean carbon stock 522.79 mean forest carbon density in the Maasim Sarangani province. Moreover, Rhizophora macronata contributed most of the carbon stock among all species collected and the most abundant in all species in General Santos City, wherein Soneracia alba also contributed the high carbon potential in municipality of Maitom, and similar to municipality of Maasim, dominated by Suneracia alba and Rhizophora macronata, which contributed the most carbon stock among all species. The findings of the result were contributed by the pools from above ground and below ground. The total ecosystem carbon stock in 4 carbon pools contributed to live trees 33%, below ground 49%, soil carbon 18%, and dead wood 0%. General Santos City, specifically in 3 selected barangays, Bula, Baluan, and Buayan, contributed in terms of mangrove species and carbon stock. A total of 6 species from 4 families were identified during the assessment. The mangrove species diversity is very poor. On another side, this study provides important analysis for elucidating the carbon stock in above ground and below ground biomass of mangrove forests in General Santos City, South Cotabato. Rhizophora macronata have been the most abundant and contributed a huge amount of carbon stock. The total carbon stock of General Santos City with 485.18 mean forest carbon density, which include the above ground biomass 33%, below ground biomass 49%, deadwood 0%, and soil 18%, resulted to the overall percentage of the carbon pool biomass. Based on the result of the study, the researcher recommended the following for the conduct of the future studies. Be in part with a rehabilitation program that helps in mangrove vegetation. Elevate awareness together with the local sector's programs about keeping the status of the mangrove. Volunteering with local sectors in assessing the carbon storage in South Cotabato concerning climate change. Engaging with activities or programs of local sectors regarding the restoration of carbon stock in the mangrove forest. This information aids future researchers in the mangrove assessment. It is important to make a double effort in raising different activities that would help in restoring and improving conservation efforts of the mangrove ecosystem in the area.